Okay, let's get started. Again, as a reminder, this presentation will be recorded and live streamed on YouTube. My name is Josua Aponte-Serrano. I am a PhD student at James Glazier's lab, and I will be talking about how to model chemical fields and diffusion using CompuCell today. Just as also as a reminder, you can find the slide deck I will be using is gonna be it's available in the Google Drive, and you can also find copies of all of the examples that I will be discussing today in the student demos folder in the same Google Drive as well. Just in case you fall behind, you can always refer back to the code. Okay, so this just for context, we are in this is module 2.3, chemical fields and gradients, the part 1. This is the second module of the day, and then I will be continue discussing chemical fields in the next module. So the first part of the in this module, I will be talking about what the biological ideas are behind chemical species, concentrations, and transport, and how this can be and how this can be thought of at least from a mathematical standpoint. Then we will continue talking about how we can then implement in CompuCell the concepts of diffusing and transporting molecules. And we will explore different conditions that lead to different profiles or concentrations of the different profile concentrations of these fields. Oh, by the way, I forgot to say, I'm gonna have to use my local version of CompuCell because I was having a little bit of problems running the demos in NanoHub, but I've uploaded the demos as zip files that you will be able to upload yourself and execute on NanoHub. I made sure that they were working, and they indeed are, but I was having a little bit of problems running it myself, so I will just use the local version. I'm gonna stop sharing for a second so I can share my full screen now. Tarun, I'm gonna have the split screen so I can both walk through the presentation and the code as well. So, so the basic idea of behind fields is that we want to model we want to be able to model the transport of molecules across or between cells or in tissue. And this is important, right? Because molecules they are essential for the functioning of biological organisms. They control the transport of such molecules, control the important processes, including cell division, morphogenesis, development. They have roles in the transport of metabolic resources, of signals, and of other cellular products. So it's a fundamental component of the biology of cells and tissues that molecules are transported in or between them. So even though even though molecules are individual particles, we can make an approximation when we assume that these molecules are relatively high in that we have relatively many of these molecules. And instead of talking about individual particles, what we made an approximation and treat them as concentrations or densities, which means that we, instead of focusing on a particular molecule and the movement and motion of that molecule, we consider the number of particles that occupied a particular space or a particular area or volume, and area or volume depending on how we think about the domain of interest, and how the concentration of those molecules change in between different parts of that space. So again, the idea is even though molecules, the interactions between molecules occur at an individual level between one molecule and another, when we can make the approximation of treating them as densities, because we assume that there is enough of them such that we can treat them, we can focus on on the number of particles that occupy a certain volume or area on the space. So there's different transport mechanisms that lead to these particles or particles moving from one area or the other, or the concentrations changing in time. For the purpose of this workshop and in CompuCell, we focus mostly on diffusion as the main transport mechanism, although there are other mechanisms that are also important and play a role in transport of molecules in biological tissue, including advection and active transport. But again, the most important mechanism of transport that we will consider here is diffusion. And diffusion is a process in which molecules move from areas of high concentration to areas of low concentration due only to their random molecular motion. So this is important because because in principle, diffusion does not require an external source of energy, except, let's say, the average temperature, right? But it doesn't require an external source of energy for the particles to, or the concentrations to change in time. So this is this is a main mechanism of transport that we will consider when modeling transport of particles in CompuCell. It's important to notice as well that diffusion, the concept of diffusion can be applied not just to individual particles, but it's an idea that it can be also applied to populations of bacteria, animals in the environment, heat, and different types of fluids. And although the underlying physics of all of these types of transport are different, the basic physics and the way to describe them are the same. So in cell biology, the diffusion is an important transport mechanism because precisely because it does not require cells to invest energy. On the other hand, diffusion is limited to transport over short distances, and this is and we will see this later on as we explore the mathematics. But it's important to, just as an idea, to understand that a diffusing molecule, the distance that it travels is proportional to the square root of time. So if it's just due to diffusion. So, for example, if a molecule takes 1 second to travel 1 micrometer, then it will take 4 seconds to travel 2 micrometers, and 100 seconds to travel 10 micrometers. So it becomes less and less efficient over longer distances. So again, that's the basic idea. So why do we? The idea is that we look at molecules. Molecules are important to model in a cellular and a tissue context, and we can make an approximation of treating them as concentrations when we assume that we have a sufficient number of them. And then what we care about is how the concentration changes from different regions in space. And we assume that these changes in concentrations are due to the diffusion of these particles, moving from higher areas of concentration to lower areas of concentration. Okay, so now, how does the idea, now that we have that idea, biological background settled, let's see how CompuCell implements, how these ideas are implemented mathematically and computationally in CompuCell. So let's just define the concentration at a particular voxel or pixel. So, or as a number that determines, let's say at a pixel determines what the concentration is over that voxel or volume or area. So once we have defined that concentration, right, which means how many molecules occupy that volume or that space, then we can define the change in concentration as proportion as the using the diffusion equation, which has which is proportional to the gradient of the concentration, and the proportionality constant is what we call the diffusion coefficient, and this is going to be important. We're going to explore later on what the effect of the diffusion coefficient is on the transport and the change in concentration. But again, the idea is that if you look at a particular area of space, how the concentration in that space is going to change in time depends on the diffusion coefficient of the chemical that we're looking at or we're interested in, and the gradient of and the gradient of the concentration at that particular space. And it's important also to note that, and this is important, we'll see it later, that the net effect of diffusion is to erase the concentrations differences across the domain. So for example, if we start with a, let's say we pour a drop of ink on water and we just let it settle there, we will, if we come back later, we would expect to see the ink to be uniformly mixed in the liquid, and that is due to the diffusion of the ink molecules. So what happens is that the differences in concentration when I put the drop in a particular domain were erased, were ended up uniformly distributed because of diffusion moving the molecules from higher areas of concentration to lower areas of concentration. And an important biological note here is that diffusion is not this if we look at a particular, let's say, tissue, we would not expect the diffusion constant to be the same everywhere in the tissue. Diffusion is faster in water than in the cytoplasm, because the cytoplasm it tends to be a much crowded space. It's usually faster in water than across a membrane because there's obstacles for the molecules to cross, and it's also faster in water than in other areas where the molecules might stick to. For example, if they stick to the any components of the extracellular matrix, or if they bind to any let's say cell surface protein. So, okay, now the idea, how do CompuCell represents this concentration? So if we imagine a two-dimensional lattice, the way CompuCell is gonna represent the concentration at each particular site is gonna be a scalar number. And that number determines what the concentration of the field or the chemical is at that particular location. And then the CompuCell diffusion solvers are gonna be in charge of solving the diffusion equation that I just showed previously to determine what the concentration, how the concentrations at each of the locations of the simulation domain changes in time. And there are multiple of these diffusion solvers in CC3D depending on the particular problem that of interest. Some of them might be more useful than others. I'm not gonna go into the details of this. If you are interested in learning about the different diffusion solvers, the documentation is here in the slide deck. We will focus on using the diffusion solver FE for the purpose of the examples in this module. So one last thing that it needs to be specified for solving the diffusion equation is what happens at the beginning and at the boundaries of the simulation domain. So at the boundaries, we have several options, and I'm explaining this conceptually now, but we will see examples of these in a minute. So there's basically three types of boundary conditions that we can have. We can have periodic boundary conditions, which is this example here in the middle. And in that case, we imagine that if a particle moves, let's say on the left edge of my simulation domain, it appears back on the right edge of my simulation domain. And we call it periodic because we imagine that the simulation domain that we're looking at is just a small, portion of a much larger simulation domain that are just copies of the same domain that I'm looking at. So that is why if one molecule disappears on my left side, one appears on my right side, because it's basically two copies of the same simulation domain, one side, one next to the other. Another option that we have is constant flux. And in that case, we can imagine that there are particles always coming in or out from the borders of my simulation domain. And our last one is a periodic boundary conditions. And what we imagine there is that there is always a source of molecules at our edges, that the value of the field at our edges is always the same. And then particles diffuse in or out based on the concentrations at those edges. And we will use this in the next couple of examples. It will become more clear as we walk through the examples. So let's just start with exploring what the effect of this boundary and initial conditions are. In this example, I will create a simulation with just one. I will show you how to create one field, how to set the diffusion coefficient that we already explained, and we will explore what happens when we start with different initial and boundary conditions. Yes, AJ, go ahead with your question.
My question was, if you consider the boundary more like a wall where the molecules bounce back,
There
what
is an
would,
option, yes.
yeah, what would be the appropriate boundary definition for the boundary condition in that context, so that it's not like it's fixed, it's not like it's wrapping around, but it's just simply like it bounced back, so that the
Right.
boundary does have an effect. It causes things to accumulate somewhat over there.
Right. So as James and T are answering in the chat, we call that constant flux. Oh, constant flux set to 0 or 0 flux.
Okay.
So there is no flux across the boundary. That's basically setting a wall in which molecules bounce back. I'm sorry, go ahead.
When your boundary is a wall, should that not be no flux condition?
Yeah, that's the 0 flux or no flux condition, yes.
So no flux in your case is constant flux set to 0, is that right?
Yeah, so that's the way we implement it.
Okay.
Just 1 second, my earbuds disconnected and I can barely hear you. So just 1 second. So we call it no flux, right? Or 0 flux or constant flux 0. But yes, that's the way we implement the basically the wall.
Perfect, thank you.
So with this example, what I want to show is what the effect of these different initial conditions are, and the boundary conditions, and then I'll show you also how to set how to set a chemical field and specify its boundary and initial conditions. So what we'll do first is we will create a new project. And we're gonna use XML, the Python XML. We can leave everything else. This part's general simulation properties have to do with what happens to cells. And since we're gonna be dealing just with fields, we don't really have to specify here anything in particular, except maybe the simulation domain. We can make it shorter just to make the simulation run faster. But we will have no cells, so we don't have to worry about specifying anything here. Same in the cell type specification. We're not adding cells at the moment, so we can hit continue. And then let's introduce one field in the chemical fields. Let's call it oxygen. And as I mentioned, we have different options of what the diffusion solver we want to use is. Those depends particularly on the problem of interest. For this, because we're going to be using relatively small diffusion coefficients, we can just stick to the regular diffusion solver. And then we hit add. And we don't need anything else. So we can hit continue. And then we will have one empty simulation that just has a chemical field. So let's walk forward. We add the chemical, we add the name. We can add the name of the field, select the diffusion solver, and we can hit add, and then we can add it. Pedro, in that particular case, right. And we can come back to that because this is an interesting example, and I can show there is an option for in CompuCell, for example, to set what the initial concentration of the field is, and then depending on what if you do not want your field to diffuse, for example, you can set the diffusion coefficient to 0, and the field won't diffuse, but you can still interact with the field depending on what your interactions of interest are. So coming back here, we add a field. We click click next, and now we in the XML we have the options to specify what the diffusion constant is. And remember, the diffusion constant is the proportionality constant that determines how the concentration changes in time at a particular rate. I'm sorry, one second. So the other parameters of the diffusion coefficient the other parameters of the diffusion equation, there's other parameters of the diffusion equation that we could also specify here in the XML, and we haven't discussed them yet, but we will introduce them later. But here, for example, you can also, there is also options to specify what the initial concentrations are based on either a formula that is dependent on X and Y refer to the positions in the plane, or you can import a file that specifies what the concentration is at each of the locations of the simulation domain. And then you can also specify what happens at the boundary. These are the options that I was talking about earlier. You have the periodic boundary conditions. You can also have a constant value at the boundary, or a constant flux or constant derivative. So, right, and depending on what our problem of interest is, we can specify our conditions accordingly. So now that we have created this simulation, let's just explore what happens if I start my simulation with a uniform concentration of 1 everywhere in my simulation domain. I am setting my decay constant to or I can set it to 0 because I am not using it at the moment. And then I'm gonna also specify periodic boundary conditions. So I'm gonna launch the poll just to make sure that everyone is if people are following. So all I've done is I created a project. I added a field that I called oxygen, and I didn't add anything else. And now I came to the XML where I can specify what the diffusion coefficient is. And I'm also specifying my initial concentration to be 1, which means that the concentration is going to be 1 everywhere in my simulation domain. And then this equation data we'll see later. So we don't need it at the moment. But then on the boundary conditions, I'm setting them to be periodic. So I'm gonna give everyone a little bit of time. This is important because we will be repeating this same exercise over and over of creating the field and coming and specifying what we want our diffusion parameters to be and the boundary conditions to be. Also, as a reminder, since this is exercise 2.31, I have an uploaded version of the file in the Google Doc, in the Google Drive I would recommend having it open as a reference. Still work through the exercise, but if you have a question, you can always refer back to that document. Okay, so quick question. I am starting my simulation domain with my concentration being 1 everywhere. That's what I've set up in my initial concentrations, in my initial concentration, or as my initial condition, and my boundary being periodic. What would you expect with, or what do you think it's gonna happen once I run the simulation in terms of the concentration? Right. Nothing is gonna happen. And why is that? Oh yeah, so that's the example. I'm sorry. I'll come if you ran my code has the next my code has the next part of this exercise also, that it initializes basically a drop in the middle of the simulation domain. So how do I boundary conditions? So if once you've added, you come to the XML file and you can find all of the chemical field information is always going to be contained in this table called Diffusion Solver FE. And then in that Diffusion Solver FE, you'll have the name of the field that you've just created. You'll have the diffusion data that contains the parameters, the diffusion parameters, the secretion data that we're not dealing with right now, so everything is commented out. And then you'll find the boundary conditions here. And then all I did was I commented out the constant values and set them periodic both on the X and Y axis. So, right, good point. So since we didn't create any cells, but once we create a simulation, the CompuCell it's going to load a steppable for initializing cells. So we need to get rid of that if we don't want any cells to be initialized in our simulation. So that's important. In this file as well, please comment out the uniform or blob initializer, so we don't have any cells at the beginning of the simulation. Okay, so now what's going to happen, as some people commented out, is once I ran this simulation, I shouldn't expect anything to happen because diffusion depends on, right, is proportional to the gradient of the concentration. But if the concentration is uniform, there is no gradient. So there won't be any change in time of concentration time. So that's what I should see. Another thing to point out is once I execute my simulation, CompuCell always executes, opens a cell field, opens a simulation in the cell field window. If we want to see the field, we come to this projection pull down menu, and then we select the field of interest. And then I can continue running my simulation. And then as people commented out, commented, you shouldn't expect to see anything happening because we didn't have any, because the concentration is uniform across my simulation domain. Okay. So this was just, so that is in initial, so in diffusion data, you have the option to specify what your diffusion constants and decay constants are. And then you have additional options to specify the initial concentration. And you also have the option of uploading a file that has, let's say, what the concentration is at each domain of the simulation. What I did is that I set it, if you set it to 1, the initial concentration expression, that means that the concentration is gonna be 1 everywhere. Okay, so now let's look at the next example. In the next example, we set the concentration to be, instead of 1, we're gonna set it to be this expression here. Well, it's 256 if your simulation domain is 256, but I set it to 100. So, because what we want is to place a dot in the middle of the simulation domain. Basically, we're simulating adding a drop of chemical on the surface of the simulation domain. Okay, I'm gonna share the poll again. It's 10 minus. So again, what I'm playing with here is the idea of starting with different initial conditions. In the first initial condition, I started with that uniform concentration across my simulation domain. Here, I am starting with a different concentration at the center of my simulation domain. Again, it's 256 if your simulation domain is 256, is 100 if your simulation domain is 100. You can always come back and check what the dimensions of your simulation domain are in DITSPOTS data. Okay. So you can see the expression there. No, thank you. Thank you for the correction. So now, quick question. Well, I mean, the answer is there already, but how do you, what do you expect to see, and what explains what you will see in the simulation, or what you see in the simulation? So what's happening? Someone can guess? So if I execute my simulation, right, basically the drop quickly diffuses and the concentrations diffuses across the simulation. So, Okay, so I was going to explain this later, but I would just explain it now. So there is a way, if you're not seeing any changes in the field, you might need to adjust your viewing options. And I'll come back to this later. So, right. So Ben, I was having that same problem with Nano, running this particular file in NanoHub, and that's why I'm running it locally. I'm not exactly sure if it's TJ might be able to help here, but I think it might be a configuration file that is really important. But I was also having that same problem that my mind was also crashing. So right, I start my simulation and I start with a dot in the center of my simulation. And as I run the simulation, I can see how the molecule diffuses away from, or the concentration of the field or the chemical is diffusing away from that original drop. The color bar, that is an excellent question. I will come back to this later, but there is the color bar automatically adjusts itself in the default options. So red is always going to be the area with the higher concentration, and blue is always going to be the area with the lower concentration. If I come to options here, I can fix what the color bar values are going to be. Let's set it to the minimum to be the minimum value being 0 and the maximum value being 10. And then I can see now that the values were readjusted and the heat map is not readjusting to the maximum and minimum. So if I start it over, I can see my dot at the beginning, but then it disappears because it's basically uniform. It has basically been uniformly diffused in the simulation domain. Okay. So again, I have copies of these files in my in the folder. In the folder, you can see in the file that I added to the folder, you will be able to see both exercises that I've done so far. If you comment out this line, you'll start with the uniform concentration. If you comment out this line, you'll start with the high concentration at the center of the simulation domain. Okay. So let's try now a little bit of a different exercise. So instead of having initial and boundary conditions, instead of playing with the initial conditions, Let's now play with the boundary conditions. And what we will do is that we'll comment out the initial concentration expression. And if I comment out this, my simulation will start with 0, with the concentration of my field being 0 everywhere. And now what I want to do is that instead of having periodic and boundary conditions on the X and Y axis, I want to start with a constant value of 1 in both my X and Y axis. And let me explain what this syntax means. So constant value plane position mean means the minimum on that plane. So in this case, 0. What is the value of the field at the boundary? 0. Constant max, plane position max, means the maximum on that plane. So in this particular case, 250 or 100, whatever the simulation domain was. Then same with the y-axis. I set the constant value to be 1 in the minimum and 1 in the maximum. Then if I restart my simulation, so I was having this problem plenty of times. If you have different boundary conditions, it's gonna start with the last one you specified. So you have to make sure that you comment out the periodic boundary conditions such that when you execute the simulation, it only has the constant value. I kept having that error, running into that error earlier. So now my field is diffusing, but the reason I don't see it is because I played with I adjusted the maximum and the minimum of the fields. So I have to readjust it so I can actually see what's happening in my simulation domain. And I set it to 1 because 1 is the maximum at the edges. So again, I can rerun this simulation and you can see how the molecule diffuses inward from the boundary to the center of the simulation domain. Do notice though that in this particular case, the maximum value that the field is going to have, it's always gonna be 1, right? The value that I specified at the edge of the simulation or at the boundary of the simulation. I'm gonna stop here for a second and give everyone time to catch up. So if it's only diffusing from one side, make sure that you specify constant value min and max on both X and the Y planes. To get to get the simulation domain to look like mine, remember that you have to change from the self field to the oxygen. And in the settings, you need to let's just, for now, eliminate the fixed values of the field so you can actually see the fields. The concentration is gonna look different based on how you choose the heat map to look like, right? You can, here in this particular example, these are what we call floating. So the minimum and the maximum on the heat maps are always gonna readjust and adapt to the maximum and the minimum of the field in the simulation domain. If instead I want them to be fixed, I can go to settings and fix them such that the colors are fixed to the values that I specified. So Tony, just again, make sure that you have specified values of the planes in both X and Y axis. And just so everyone remember, I have a copy of this particular file for reference. And it has all the options that I've played with. I've just been commenting stuff in and out so you can all see the different condition sites I've specified. Right. So you need to make sure that you're adjusting the constant value on both X and Y axis and not the constant derivative. Right? Because remember, constant value is one type of boundary conditions where the value of the field is always constant at the boundary, whereas constant derivative is a different condition that we will see next. Yeah, I'm gonna give people some time because this is the end of exercise 2.31, and I want everyone to at least get some familiarity with it. So again, just as a reminder, once we've specified the field in CompuCell and we open the XML, we will see this diffusion solver steppable that will contain the data of it's going to contain all the information pertaining the chemical field. The first part is you're gonna specify what the diffusion coefficient, the diffusion data is, the diffusion coefficient, the decay, and the initial conditions. And then you have another section where you just specify what the boundary conditions are. Right, I commented it out for this particular simulation, for the simulation when I'm starting with a constant value at the boundaries. Cato, do you have a question? How to fix the lookup table? 1 second. There you go. So that is the link to these files. And you can, the exercise I'm looking at is the 231, exercise 231. So again, I'm giving everyone time because all the other exercises in this and the next one depend on we understanding how to specify our diffusion data in the XML. Yes, so again, I'm gonna redo everything I did quickly because I can comment out stuff here. So the first simulation I started with so the first thing I did after loading the XML was that I set the decay constant to 0 because we're not going to worry about decay right now. We just want a diffusing field. And the first simulation that I did, I specified my conditions to be periodic at the boundary and my initial concentration to be 1 everywhere. And when I do that, what I see is a uniform field that nothing happens because there is no gradient in my simulation. Okay. The second example, instead of starting with a uniform concentration everywhere, I started with a dot or a drop in the middle of the simulation domain. And then what I see as I run the simulation is how that drop diffuses from the center of the simulation domain, where I placed it at the beginning. Okay. The next example that I showed was what happens if instead of having some initial concentration inside the simulation domain, I set a constant value at my boundary. And I noticed people were having this problem. I have that problem all the time too, that I forget that it has to be, the tag in the XML has to be constant value. If I specify constant derivative, that is gonna do something different. So instead of having an initial concentration, I started with some value, some constant value at the boundary. And in that case, I can see the field diffusing from the boundary inwards. Yes, Ford. That is, to do that, you to readjust the color schemes, you can come to settings, and then it's gonna allow you to adjust what your maximum value and your maximum minimum is gonna be. And remember, your maximum value is always gonna be red and the minimum is always gonna be blue. Okay, and then the last, now I'm going to continue with the last part of the exercise, which instead of having constant value, we're going to have different boundary conditions with the constant flux or constant derivative at the boundaries. And then when I run that simulation, I can again see the chemical diffusing inwards from the edge of the simulation. But now we can notice that the maximum of the field, instead of being 1 as in the previous example, it's always increasing. Anyone has a guess as to why is that the case? Right, because constant derivative is the same as a constant flux. And that's an excellent question, and the answer is the latest. What the numbers here mean are gonna depend on how you're interpreting the values of the field. And generally you're free to interpret them as you want in terms of let's say, as a concentration, number of particles, and so on. But yeah, you have to be consistent once you've said, for example, what the length of your pixel is. Okay, so yeah, and so I'm gonna answer the question here. The reason we are seeing this number continue to increase as opposed as in the previous case where it was constant, is because in this case we're constantly supplying to the simulation domain from the edges, right? So the concentration is always gonna keep increasing because we're basically always supplying from the edge. Whereas in the previous one, instead of supplying, we had just fixed the value at the edge and let the molecules diffuse inward. But in this particular case, we are sort of like supplying in from the edges. So that's why it keeps increasing. And that is the difference between having a constant flux and a constant value at the edge of your simulation domain. Okay. So now that we have explored our different options of initial and boundary conditions, let's look at what happens if we try to solve we solve the diffusion equation with a fixed boundary values and no decay. So in that particular case, we can solve the diffusion equation. This is a diffusion equation quite easily. And the solution of the diffusion equation is gonna be this. It's basically saying that the concentration is gonna change linearly on the position. And that the equilibrium concentration is independent of the diffusion coefficient. So for example, if we start at one boundary with a value of 100, then the diffusion coefficient, the solution to the diffusion equation with no decay is gonna be this equation here. Of course, when we start the simulation, depending on the initial conditions, we will not necessarily reach this equilibrium concentration. But if we let the system run for long enough, the system will eventually equilibrate to the solution of to the solution of the diffusion equation. But the time that takes for the system to equilibrate to that solution depends on the diffusion coefficient. And that introduces the idea of the diffusion time. And the diffusion time is usually the time that it takes a molecule to travel a distance of a distance, let's say L. So you can see that the and this is what I was explaining earlier, the time that it takes increases as the square of the distance. So if it takes 1 second to travel 1 micrometer, it's gonna take 2 seconds to travel 4 micro. It's gonna take 4 seconds to travel 2 micrometers. It's gonna take 100 seconds to travel 10 micrometers. So diffusion is effective over short distances, but it slows rapidly over long distances. And that is why, for example, cells take advantage of diffusion over short ranges where they don't have to invest energy. But if they want to transport molecules across bigger distances, they generally have to rely on active methods of transport that require using energy. So now what we will do is that we will specify, we will see how we can implement this linear gradient or this linear concentration using CompuCell and playing just with our boundary conditions. And what we will do is that we're gonna write a simulation where we impose a constant concentration on the left side and a 0 concentration on the right side. And in this particular example, we're also gonna learn how to measure that gradient and how to plot it. So when we create this simulation, we have to remember that we want the diffusion constant to be uniform everywhere. So we won't have cells. We want no decay. And we want and we also want fixed value on let's say the X and Y axis, on the X axis, and then boundary conditions on the Y axis. So let's try to get that, let's try to generate that simulation ourselves. So we come back to, I'm gonna relaunch the poll. Okay, so we start a new project. Oh, and just for reference, again, this is example 2.3.2. But if we want to start from the beginning, we create a new simulation. I'm going to call it field 2. And I'm going to use, just because it's easier for me to think in powers of 10, I'm going to use simulation domain of 100 by 100. I'm going to have no cell, and I'm going to add an oxygen field. And then I finish with my simulation. And then I can come to my XML. And again, I have to specify the properties of the chemical field in this diffusion stepable. So let's set the global diffusion coefficient to be 1. We are not interested in our molecules decaying at this moment. We'll come back to that idea later. And then I said that we want in the x-axis, we want our value to be 10 on the minimum and 0 on the maximum. And then on the y-plane, we want our conditions to be periodic. And again, remember, every time we create a simulation, but we do not want to have cells, we have to delete the initializer. Okay. And that's all I need for this simulation to run. I want you to give it a try and let me know if it's running on your case and what you see when you run it before I move on. Seems like most people caught up with this exercise. So can someone tell me what they're right? So now you basically start with red on the left side and or the chemical on the left side, and it moves to the right side. So I'm gonna execute the. Remember, to see the field, I always have to come to this pull down menu, and then I can execute the simulation. And I can see again, so for example, here, I can see that the numbers are, that the heat map is, it's free floating because the minimum and the maximum are adjusting themselves. So if I want to fix them, I can come to my settings and I know what my maximum is, right? Because it's coming from my left edge, and that is 0. And I know what my minimum is because it's the value that I established at the right edge, and that is 0. It's the maximum is 10, the minimum is 0. So I can adjust that, and I should be able to see my gradient uniform or linearly distributed in my domain. Okay. So this is what I was explaining earlier. If you want to adjust what you see, you can do it in settings. So for example, if I set my maximum to be 1, then my field is gonna look different because everything that has a value of 1 or bigger is gonna be painted red. So that's why it's correctly interpret visually this simulations, you need to be very aware of what your display settings is. And there's another visualization option that we have where we can show lines. So if we go to settings and we click on show the scalar contours, this is gonna draw lines that show regions of the domain that have the same concentration. So make sure that your simulation is running. To make sure that you can, you need your simulation to be running in order for it to adapt to be able to fit, to play with the settings, visualization settings. So again, these contour lines show regions of my simulation domain that have the same value of the concentration of the field. And I can play with how many of these lines I want to show. Okay. So we call this linear gradient, right? Because the field is linear, it decreases linearly from one edge of the simulation to the other edge. And that's why these contour lines are equally spaced between each other. Now, we can also, yes, absolutely, Tony. So all I did in the code was I set my decay constant to 0. I set in the boundary conditions to be constant value on the X plane, 10 in the minimum, 0 in the maximum, and periodic on the y-axis. Okay, and I'm going to show you now how we can measure and plot the concentration, not just look at it. I'm not necessarily sure if we cover plotting already. This is going to be a refresher. If not, this is probably the first time we'll see plots, but this is also very important. For plotting, we always do our plots in the steppable in our Python steppable file. We've been so far playing mostly with the XML, but if we want to add scientific plots to our simulation, we do that in our steppable files. Once we are in our steppable files, if we want to have access to if we want to have access to the code that would allow you to generate these plots, we come to this pull down CC3D Python pull down menu. And we come to scientific plots. And there's a couple of options here. We have a setup option and an add data point option. And then we have this erase plot option, which we will be using. So if the first thing we need to do is to set up our scientific plot window. And we do that by generating this code snippet inside the start function of our steppable. When we click on it, we get this code. The first line of the code basically creates the plot window, and it has as properties a title, an x-axis title, and y-axis title, and then whether we want our x and y scales to be linear or log scales. We talked about that yesterday. So what we will do is that we will call our plot, we will call it concentration versus or per position. Our X title is going to be the position, and our Y axis title is going to be concentration, and we want them to be linear. And then the next two parts of the code snippet create some data series that are going to be added to this plot window. Since we just want to measure the concentration, we just want one data series, and let's call it CON. And we can specify what style we want. In this case, I'm going to have dots, but it can also be a line. and the color and the size of the markers. And if I execute my simulation now, I'm sorry. If I execute my simulation now, I can see that I now have a concentration per position window and a plot. But as I execute the simulation, I don't get anything, nothing is being plotted because I haven't added any data to the data series. So to add data to the data series, I can come back again to the CC3D Python scientific plots, and I can add data point, and this is going to be inside my step function. And the code snippets suggest this syntax. It's important to match the name of the data series to the one I created in the start function, right? And then the second argument is going to be what is going to be plotted on the X position, and the Y, it's going to be what is going to be plotted on the Y position. So if I want to measure and plot the concentration, conceptually what I have to do is I have to iterate through the X, let's say on the X plane, and measure what the concentration of the field is at that particular location, and then plot that location and the concentration. So let me just relaunch the plot to make sure that everyone is following. Okay, good. Okay, good. So the way I do that is I'm gonna iterate, I'm gonna create a loop where I iterate over my X domain, and I do that. I have this property called self.dim.x that gives me back the length of my X domain. And then I can add this inside of, I can move this add data point inside of this loop. But now I need to basically get or fetch what the value of the concentration field is at that particular location. And to do that, I have this, I can come back to my CC3D Python pull-down menu, which is gonna be your best friend. And I come to chemical field manipulation, and I need two things. I need a reference to the field, get field reference. And I also need to get the field value. So now that I have those two things, I can, so I know I have 1 more minute left for this module. I'm gonna run over until 3:50, so I can finish showing how to plot, because this is important, how to measure the concentrations, because this is important. And then we'll continue with the next module after. So for referencing the field, all I need is to tell that field that, and then I have to change to the actual field name as created on the XML, which in this case is oxygen. And then to fetch the value, I just use this syntax where I specified the field position that I'm of interest. So what I can do is I can move this inside of the for loop. And then ask the question, sorry. And then I can ask the question, what is the value of the field at position X? And I also have to specify Y. It's not important in this particular simulation because Y is the same value across the Y domain. But if it wasn't, I would have to specify what position in the Y domain I'm also measuring my concentration. And in this particular case, I am measuring halfway of the simulation domain. So now I can pass on this value to the add data series. And then because I want the plot window to show how the concentration changes in time, I need to after I plot, I need to erase the data and then basically plot the concentration again. And the way I do this is I can come to CC3D Python scientific plots and erase plot, and then it's gonna, that's also added in the plot function, in the step function, and make sure to add it before your for loop. So this is important because we will also come back to this in all the other examples. And now if I run my simulation, I can both see how my field is, my chemical is diffusing in the simulation domain, but I can also see how the concentration is changing at each of the locations of the domain. And at equilibrium, I should expect this to look like what? Since it's called a linear gradient. So it's supposed to look like a line, right? Because and that is why we call it a linear gradient, because the concentration is decreasing linearly from the source to the sink. Since we ran out of time, what I wanted to show next was what happens if we, and we will come back to this, but what happens if we, and I'm gonna start this one next model here because it's important, but just as a teaser, what happens if we change the decay that we had specified it to be 0, if we instead change it to be, let's say, a non-zero value, then the profile of my concentration changes, and instead of decreasing linearly, it now decreases exponentially. And we will start the next module with this example, but this is just as a teaser, because I wanted to show you what the difference between having a linear gradient is at a linear and an exponential gradient is. And we can see that also how long does it take to reach that equilibrium value depends on the value of the diffusion coefficient. Yes, the Pi file it should look like this. And this is important because we will continue using this code, this part of the code for all the next couple of examples. And I'm gonna stop here and I will come back and start over in this part of the module. In 10 minutes. Please do feel free to ask questions while in the meantime, if you need some time to catch up. Oh, okay. Hold on. I'm gonna share my screen again, my full screen, so everyone can see what the plotting looks like. But again, remember that all of these options are available for, you don't have to know them from the top of your head. You can always reference them from the CC3D Python pull-down menu. Remember, we use scientific plots, the setup, the add data points, and the erase plot. And we also use the chemical field manipulation. We use the get field reference and the get field value. And the reason I need both is because I need my Python code to be able to reference back my XML code. And that I do by getting this get field reference.